So now we're going to finish up with the last two of the four carbon-containing molecules of life. So we're going to talk about proteins, and that's supposed to say nucleic acids, not amino acids. But all right, so talking about proteins. So proteins, again, are a polymer. So it's a bunch of little monomers bonded together. So it's a polymer of what are known as amino acids. So each individual amino acid is the monomer, and then the protein itself is the polymer. So there are 20 different amino acids. Okay, there are some modifications that can happen to these 20, but for the most part, those are relatively rare. So it's considered that there are just 20 different amino acids. Don't freak out. You don't have to memorize these. You don't have to draw the structure or anything. If you go on into biochem, generally you will have to. It is kind of useful once you get up into the higher level proteins and all of that kind of stuff. But for just general biology, we don't need to know that. What I do want you to know is the different types of amino acids that are possible to find. So you've got your nonpolar. Okay, so these are the ones that are not going to want to contact any polar molecule like water. Okay, so they form very different interactions with each other. Then you have polar molecules, so these love being right next to water, but they don't like being next to nonpolar things like these amino acids. Okay, and then you have your positively charged amino acids, each of which will have a positive, it turns out that they're all nitrogens. Okay, so each has a positive nitrogen. And then you also have your two negatively charged amino acids. And you'll notice that both of them are negative on oxygens. Okay? So, 4 different types of amino acids, or 4 general types of amino acids. There are 20 different amino acids. Okay, this has all of them here. Yeah, they kind of have some weird names. Arginine, tryptophan, phenylalanine. I actually like that one. It's fun to say. Phenylalanine. Anyway, yeah, so 20 different amino acids, 4 general types of amino acids, and then the amino acids differ in their side groups. These are known as R groups. So if we look back at all of these, you'll notice that this top part here, the part that's not highlighted a different color, it's the same in every single amino acid. Okay, so that is actually what gets bonded to each other. All right, so it's this negative oxygen here would bind with the positive nitrogen on the next amino acid. Okay, and that's how they form these long chains. So the only thing that's different between each amino acid is what's bonded to this first carbon atom here. Okay, they can be something as simple as the glycine, which only has a hydrogen bonded to it, to as complex as tryptophan that has a double ring structure on it. Okay, or even arginine that's got a fairly long chain for an amino acid on it. Okay, so the side groups, the R groups, are what differ between the different amino acids. So this is just kind of showing you, again, the basic structure of the amino acid. You'll see that you have a nitrogen on one end and then a carbon on the other end with the oxygens that break off. Okay, so you have your N-terminal end, okay, N-terminal domain, or your NTD, and then you also have your C-terminal domain, your CTD. Okay, that just determines, or basically is telling you what side of the protein or the amino acid you're talking about. Amino acids are linked by what are known as peptide bonds. So that's just the bond between this carbon and this nitrogen here. Okay, it actually replaces this oxygen, right? So it kicks it out. So this oxygen and the hydrogen here are only present when the amino acid is not bonded to something. And then when the bond takes place, it takes this oxygen and hydrogen, and a hydrogen off of the nitrogen takes those out. So it is releasing water whenever an amino acid bonds to another amino acid. Right? So these are called peptide bonds, just a specific name. And fun little thing, protein is also known as a polypeptide, okay, because it has multiple peptide bonds in it. Because, of course, scientists can't use one name for something. It has to be multiple different names. But proteins can differ in the number and the order of amino acids. You can get, I think the smallest protein is about 3 amino acids long. It's a very strange protein. It's got amino acids all different kinds of ways. But longest, we don't actually know because we keep finding longer ones. So it can go quite a length of amino acids, quite a number of them. Amino acids will interact to form the protein's shape. And if you remember what we talked about in the last chapter, form follows function. Okay, so the shape of a protein is incredibly important to its function. In fact, if the protein is misfolded or is not the correct shape, generally it will not work at all. Okay, or worst case scenario, it starts doing something wrong. So then you get a lot of bad stuff happening. Okay, that's actually what we see in Alzheimer's, where you get proteins that misfold, so they aren't doing the function that they're supposed to, but then they actually go on and attach to other proteins and force those proteins to misfold, which is why you get all kinds of issues in the brain, because all of these compounds, all of these proteins are basically falling out of the brain. They're no longer useful. And it gets to the point where it basically just destroys the brain. So that's why long-term memory and really short-term memory just leave. Okay? So this is looking at a specific protein. It's called hemoglobin. This is actually what binds the oxygen in your blood, so this is a relatively important protein for us. It is actually held together through mostly hydrogen bonds. So hydrogen bonds, which we talked about with water, but the hydrogen bonds will form between different amino acids. And those amino acids don't have to be next to each other. They don't even have to be anywhere close to each other in the amino acid sequence. Okay, you can actually get 2 amino acids from completely opposite ends of the protein interacting together once the protein folds. Okay, so there is no like rhyme or reason as to, if it's close together in the amino acid sequence that they're going to interact, because usually they don't. Okay, now there are actually 4 different levels of interaction that can happen in the amino acid or in the protein. Okay, or 4 different levels of organization. So you have primary, secondary, tertiary, and quaternary. Basically, primary is looking at the amino acid sequence itself, and then when you get up to secondary and tertiary, you're looking at the different folds and shapes that arise in the protein. And then if you get up to quaternary, that's actually 2 different proteins interacting together to create one function. So for an example with hemoglobin, there are actually 4 different hemoglobin proteins that come together in one red blood cell. Okay, now this is where the amino acid sequence comes in really important. It's vital to the function of the protein, because if you have one of these amino acids that's supposed to connect through a hydrogen bond, but it's the wrong amino acid, so it doesn't have that hydrogen that's necessary for the hydrogen bond to take place, then you don't have that interaction. So you won't get that fold. So an incorrect amino acid change in a protein structure can actually completely destroy the function of the protein. And that can be just a single amino acid difference. The example we always use in biochem of that is cystic fibrosis. So cystic fibrosis is a genetic condition. It is relatively rare, but not so rare that we don't know anything about it. It is caused by a single point mutation in the DNA sequence. Now, we're going to get more into DNA sequence and all of that, but what that equates to is basically it changes a single amino acid. Okay? And that single amino acid completely deletes the function of the protein. And so then you get all different kinds of symptoms with these cystic fibrosis patients. They have mucus buildup in their lungs, in their ducts. So basically their pancreas works just fine, but can't get anything out of the pancreas. So all of their digestive enzymes aren't getting out into the intestines. So the food that they eat is not getting digested properly. So in order to kind of counteract that, they have to take handfuls of enzyme replacements, pills, with every single meal. And so that gets kind of expensive and, rough on their life. And then I actually had a friend in college, or have a friend in college who has cystic fibrosis. And shortly after I graduated, he ended up with pneumonia. Now, where pneumonia would suck for a healthy person, with a cystic fibrosis patient, cannot say that tonight, because they have that mucus buildup in their lungs, they cough a lot, but that mucus actually provides a perfect growing environment for the pneumonia. And so he ended up in a coma for about 3 months because of the pneumonia. He did finally survive it and get better, but that was a pretty scary time. And just it was pretty bad. But so that is all caused by a single amino acid change in the protein sequence. Okay, so when we talk about amino acids in their sequence, you have to understand how important that is to the protein itself and to the function of that protein. So this is looking at the quaternary structure of a hemoglobin molecule. So hemoglobin, you have these 4 different chains, okay? And each one has an iron molecule bound in the center. Okay, that's what the little red dot is, all right? And then these 4 chains interact together to bind oxygen within a red blood cell. Okay, so it can bind up to 4 oxygen molecules at once. Okay? And this is showing you some of the secondary and tertiary structure of the protein as well. We're not going to get super into that. Just know that it's there. So there are those 4 levels of structure in the proteins. Okay? And then this is showing you a depiction of different molecule, and you can see these loops, these colored loops here, and spirals and all of that. Those are the actual protein chains. And then you can see the other molecules. There's a green one right here, and then the blue one here. Those are different organic compounds that are bound to this protein. I believe this is actually a different depiction of the hemoglobin protein. So, and then this is showing you kind of a space-filling molecule, or space-filling model is what this is called. So you can see that there's actually a hole right in the center of this molecule. Okay, and then you've got the different proteins interacting around that little central hole. Okay? Alright, so that is it on proteins, and the next section is going to deal with nucleic acids. So we'll leave that for the next part.